All right. So what I said, I will tell briefly how we discovered vaccination. Now going back to 1700 time, many families are self-sustained because they have farms and most of them are doing farming and they get their food from their farming. So when it comes to farming, they raise cows for milking and also get the milk and for other products out of milk. So he noticed around this house, around this neighborhood, a lady, milkmaid, the milkmaid, the lady who is going to take milk from the cows, it's called milkmaid, Sarah Nels. She's infected with cowpox. You know, cows also get similar kind of a disease from virus. They also get this kind of a skin blisters like things, which is called cowpox. But cowpox is not exactly the same virus infecting human. Cowpox cannot. Cowpox is the cowpox and smallpox. They are two different viruses. So she's infected with cowpox because he noticed there are few blisters like things, and cows also get these things. So this picture is quite informative. You can see the cow's milking place is called udder. The udder is the mammary glands of the cows, and this area very sensitive. They get this kind of blue color skin blisters. So she also got the skin blisters, and it is a contamination of the virus from the cow, and she got cowpox. Now what he did, he got this small boy, and then he just make a small scratch on the skin and transfer this content of the blisters onto the skin of this person. Skin means there's a small scratch made, and then they transfer the blisters to this place so they can go in. That means the content of these blisters can go into this boy, because we know skin is a barrier. It can't go, but we break the skin barrier mechanically, and then the content can go into this boy. So this is called inoculation. Inoculation is a common term we use, transfer of microorganism to one place to another place. Then the boy also got the cowpox, but cowpox is not as bad, just a little bit sick, a little bit ill, a little bit sick. Otherwise, he has no issues. Nothing much happened. So he got cowpox, nothing much. Then next thing, then what he did, this time, they collected the scabs. Scabs are the one with the blisters when it's dry. When it dry, the fresh blisters, it calls scabs, a bit of dried blisters. And they collected these content from a smallpox patient. Now, I will again tell you if you don't understand. Milkmaid, the lady got cowpox. Cowpox is not a serious disease. It's a mild infection and few blisters. And then he transferred this content into the body of this boy. And he just got small infection, nothing much, blisters. So cowpox is not a big problem. And next time, he got smallpox content from a patient, smallpox patient. and then place this inoculate again to the boy. So boy is challenged with smallpox. That means boy is first given cowpox contained into the body, and boy is challenged with smallpox. He's unaffected. He's protected. His name is Pips, is unaffected, protected. Now, what I want to tell you, it's very easy process for us now. But 1796, young Edward Jenner, as a medical student or a doctor, how can he think about this kind of a complex mechanism? It's only his observation and his thinking. So he thought there is some kind of protection with cowpox. Immune system make response against cowpox. This can be protected by smallpox. So this is the discovery of vaccine. 1796, this is the discovery of vaccine, and a smallpox vaccine is the first vaccine developed. As a result, the long story we had completely eradicated. Now you have another idea, what is vaccination? Vaccination is given in this picture, we can say if we given similar virus, which is not harmful, then we are protected from bad viruses. If we give viruses to the body, which is not that bad, we can get protection from strong viruses. So this is something we can understand. We exposed to diseases. We exposed to many diseases. We develop immunity against this virus. We have protection from similar kind of virus. Okay, so that is a discovery of virus. And when it comes to our case, we have two types of virus. But we learn only two. There are different vaccines. We only going to learn only two. Okay, first one, live attenuated vaccine, right? Live attenuated vaccine. Now, what is live? Live means it has a life. The cell has a life. Now, what is the meaning of attenuated? Attenuated means it can't cause the disease. Let's say virus. This is a virus or bacteria. So these bacteria virus, we consider kind of alive. That means they have a complete cell structure, or virus has the complete structure of its particles, and they are attenuated, mean they can't cause disease. They cannot cause disease. Now, if you ask why they can't cause disease, because they have modified in the lab. They are modified in the lab. Now, vaccine development is not an easy process, because scientists, they do many things. I mean, here we are not going that much detail to learn. Just word attenuated, we want to learn. Scientists do many modifications in the labs, taking this virus or bacteria until they confirm experimentally they can't cause a disease. So attenuation is cannot cause disease. So we cannot learn more than that, because if you want to learn how they modified in the lab, it's a complex process, and in our level, it is not necessary. But you can only remember attenuation is they cannot cause disease. The pathogen cannot cause disease, but they're live. They have a complete cell structure and they're live. Okay, now next thing, we inject these live attenuated pathogen into the body. Or there are other method. This is inject. Or other method is we can give a mouth, oral. If we give from the mouth, we call oral. If we can inject into the, if we can give directly into the body, it's called inject. So oral route is mouth, inject is directly into the body. Then what will happen? Go back to the next level, what we, previous class, what we learned, the immune system activate. Immune system activate from all the levels, humoral, cellular, and antibody produce, and memory cell produce. Memory cell and antibody produced. Memory cell antibodies stay in the body. So this is the method of vaccine, live attenuated vaccine. So here example, two good example, polio, oral vaccine of polio, and BCG. So I think all Sri Lankans, they get these two vaccines, polio and BCG. And I think every person, if you look at your hand, you have this kind of a mark, a tattoo. This is our natural tattoo. It's a confirmation of BCG. BCG is given for bacteria, Bacillus tuberculosis. It's a disease against tuberculosis. So the bacteria is Mycobacterium tuberculosis. So this is given at first week. Even a person, baby leaving the hospital, usually the BCG vaccine is given. This is again, this is called tuberculosis. This one again, polio virus. So this can again cause some kind of infection in the nervous system and muscles. So if person is infected with polio, person develop lifetime deformities in the legs or some areas. That's also very bad. Tuberculosis is again a big disease in the world. Theoretically, 25% of global population is infected. 25% of world population is infected with tuberculosis. But most of them, they don't show the signs. They don't show the disease because immune system is protect getting the disease out. But the bacteria is inside the lungs. Bacteria is inside the lungs. And if the immune system get weaker, then these people develop disease. So our country at the moment is quite good because of this good vaccination program compared to certain countries. We have a certain level of protections from BCG. So BCG and polio is live attenuated vaccines. Then the next type of vaccine is inactivated. Inactivated or killed. So inactivated is killed. So again, virus or bacteria. I mean, I don't want to use word for kill for virus because virus is not a living things, non-living things. But just to speak here for bacteria, yes, the virus is also inactivated. So inactivated is the best word, rather kill. They inactivate by physical or chemical methods. So scientists know what kind of a chemical use, what kind of a physical methods, what kind of a radiation or something used to inactivate them. They are inactive, but their structure is there. That means whole structure is preserved. Nothing happened to the structure. Again, we can inject to a person. So then immune system doing the same thing, just like a natural infection. They try to develop immune, they try to activate immune system, but not in a bigger way like natural infection, because they are not replicating in the body, but they have antigen. So for this antigen, they can develop immunity. So recently, we all got this Sinopharm vaccine is inactivated COVID-19 virus. Sinopharm, but Pfizer and the another vaccine people got during the COVID-19 time. They are different vaccine, but Sinopharm is inactivated virus. We got it during the COVID-19. That's a right. So example like this, right? Okay. Now we are coming to a passive immunity. Okay, if you don't see okay, the question is what if you don't see this one, right? If you don't see this one, you can check whether you have vaccine card. If you can find vaccine card issued by the hospitals and it's written your information and it has some kind of confirmation that BCG vaccine is given. If you don't find that also, you can find other parts of the body like legs, thigh area, because some people nowadays, because of this kind of a small scar here, some kind of a aesthetic reason, they can also use thigh area for vaccination. Even there's no record of this vaccine, BCG vaccine. Nothing you can do because BCG vaccine is not effective in adults, or even after first few weeks, we can't get the vaccine. It is not effective. BCG vaccine is not effective. But don't worry, people in USA, Canada, Europe, Australia, they don't do a BCG vaccine. They don't do a vaccination for tuberculosis because they have no tuberculosis, or almost very few number of tuberculosis cases. So only thing is, if you want to go to those countries, like especially USA, you have to do a special medical test for tuberculosis. Then you have to take this medical report for visa that you have no tuberculosis. That is only a possibility if it is not vaccinated. Later vaccination is not effective. Okay. Again, BCG is given, the mark is not there. Well, the mark is, it's not 100%. Like some people, it's like almost like a nothing, but some people very small, some people bigger. So some people know. I don't think this is a big problem, having a scar. So most of the people they have the scar, but some they don't have the scar. So if you have a records, I think it's fine with the BCG vaccine. Now we are coming to the passive immunity. Now, first one is naturally how we get passive immunity. Well, it's like this. Interesting thing is when the baby so this is a fetus. This is a developing fetus. The developing fetus is connected to mother side through a cord. That's a USB cord. That's a umbilical cord. This is called umbilical cord. And this umbilical cord consists of the blood vessel, artery, and vein. Umbilical artery and umbilical vein. So this is the connection to the baby side from mother. And this structure further change in mother side like this. And mother's blood supply is now coming to this place and is leaving. So this is called placenta. Mostly nutrients and the gas exchange by diffusion. There's no direct contact, it is a diffusion. So example, oxygen diffuse, carbon dioxide goes to the other side. And nutrients can also diffuse. So it's basically a diffusion method. So when all these molecules go here and there, there's another molecule which is also have privilege to move to this side. There's another molecule, important molecule, coming from mother to baby. Can you guess what is this molecule? Do you have any idea about these molecules? The molecule is antibody or immunoglobulin. You know, both names are same, antibody and immunoglobulin, same name. It is going to the baby side. So mother has a good immunity. Mother has a lot of antibodies for different pathogen. They all pass into the fetus. Then before birth, the fetus has antibody from mother. Because immune system of the fetus is not well developed. It's still trying to develop. So after birth, after birth, the baby is protected from pathogen because baby has so much of antibody from mother, and first day, first milk is also rich in antibodies. This is called colostrum. First milk is called colostrum. When the baby starts to feed immediately after birth, the first milk has very rich amount of antibodies. So antibodies is going into the digestive system. Luckily, the baby's digestive system is not really good in developing this protease enzyme because antibodies are protein that can digest. But early few days, the antibodies can go into the baby's side without digestion, and breast milk can also give this antibody. So baby is getting so much of antibodies during the pregnancy period and also after birth from the breast milk. Now the baby is protected from pathogen. So baby's immune system now start to build up. With the time, the antibody give the protection for few weeks or few months, if not sometimes few years. That's why, we say generally don't take babies after birth to every places uh, because it's not good to expose to pathogens. They can get sick. One hand is true because immune system is not the same as adult. It's developing. Immune system is developing. Protection only came from antibodies. So this kind of immune type is called naturally acquired passive immunity. Naturally acquired passive immunity. Okay, next is artificially acquired passive immunity. How we can understand this? So far we understood three. Naturally we exposed, immune system developed, fine. You can give vaccine, immune system activate. Antibodies is coming from mother to baby, then give the protection passive, naturally. Now artificial passive, artificially we give antibodies. We give antibodies artificially. Okay. Now, these days, we can produce antibody out of the bodies, right? That means animal or even host or any rabbit or host or any animals, they naturally produce antibody. If scientists or medical purpose, if we want to produce antibody, we can take animals. Animals are always easy to use for experiments than human. Then we take animal, we inject antigen, then we can purify antibodies from the blood. That's an easy technique. That's a very easy technique. Developed many years ago. They take antigen, inject to the animals. These animals develop antibodies and we purify the antibodies. Then we can use it. Now we develop this method. We don't want to use animals. We can do it using a cells. What are the cells? Plasma cells. So we can take plasma cells because plasma cells secrete antibodies. So we have to modify plasma cells using another cancer cells. We fuse plasma cell and cancer cells, and then these cells divide many times, and we use this lab technique to produce antibody. So we are not going to learn in details how precisely each step of production of antibody. But you know now, we can produce antibody. Even certain cases, we can collect blood from people. We can collect blood from people and we purify antibody. And for this case, they pool the blood of 20,000, 30,000, 40,000 people. That means they take blood from many people and they place into one container. The plasma, right? From many people, they take plasma and place into one container. Why is that? Because different people exposed to different pathogen. So they have a different level of antibodies, different type of antibodies. Now this container rich in any kind of antibodies. It's a mixture of antibodies. This is also available in hospital and some places for some diseases, as it is really good in any kind of antibodies. If we don't know precise antigen in a person, we can give this mixture, or we can call it kottu, right? Just like a kottu, it's everything is mixed here. So all antibodies are mixed, we can give. This is also available. An animal form is also available, and antibodies again available by produced by the cells in the labs. Now, why we need these antibodies? For diseases, mainly for diseases and other purpose. So we can inject these antibodies to person if we have some problem. For example, hepatitis A is a viral disease transmitted by food and water, and then the virus move into the liver, and they try to live in a liver cells. So then people develop a fever, vomiting, and so much of conditions, difficult conditions. And some people, it is very serious conditions. As soon as doctor detect the person has hepatitis A, immune system take times to develop. It's not a 1 day. It's not 2 days. It may be a 7-8 days. Immediately, antibodies can be injected because immune system finally produce antibodies. So you have learned immune system to finally produce antibodies. So we can inject antibodies to certain diseases. So then it works. Hepatitis A is one disease, and the other one is tetanus. Tetanus is caused by a bacteria, Clostridium tetani. Now, probably all of us aware, if we cut our body with a rusty object, maybe a rusty nail or knife, then these kind of places, Clostridium tetani can live. So if you have any severe damage to the body, there's a chance of infecting Clostridium tetani. Especially accidentally, we cut our leg or some places with sharp metal objects. The metal objects, we can find Clostridium tetani. And that case, maybe many of them, they have experience, ask immediately, take the tetanus vaccine. So even same day or next day, you go to the doctor to take the tetanus vaccine. Because if not treated, you can see what happened to this person's body. When it's severe conditions, which is called opisthotonus, the backward arching of the head, neck, and the spine, because it affects the nervous system. The Clostridium tetani toxin affect the nervous system, and this person is showing this kind of a body posture because of the toxin affect the nervous system. So, not a simple thing. If we are wounded and there's a suspect, there could be suspect of tetanus, it is recommended to take the tetanus vaccine. Even people have taken tetanus vaccine in the past, maybe antibody level goes down with the time. Freshly, they have to take again tetanus vaccine. Some vaccines, lifelong protecting. Some vaccine protect short period. Some vaccine protect us some years. So tetanus is something not for long duration. It is better again we take the tetanus vaccine if there is a suspicion of tetanus. So if not vaccinated immediately, I think antibodies can be given. For the treatment, tetanus antibodies can be given. Anti-tetanus antibodies.
Sir?
Yeah.
Why is tetanus vaccine is not given early when we are born?
You know, it's a good question. Okay, now we should understand in our theoretical knowledge of biology, medicine, some areas we are very weak. We don't know actually. We have the feeling everything is known now, every information available, but it's not the case. So many things are not so clear. For example, COVID-19 vaccination. Scientists, they came up with so many ideas. Number one, some are against, don't give the vaccine. This vaccine is not tested. This vaccine is not sure. The method is new. Don't give vaccine. Like even expert in the field, Nobel Prize winners like people against. And then again, should we give 1 dose, 2 dose, 3 dose? How long people get COVID-19? Can they get again this disease? We had many questions. The answer is immune system is very complicated and complex. We cannot generalize. That means certain diseases, if we get naturally, the protection is lifelong. For example, chickenpox. So chickenpox, many people they got in their school time. Because if one person got in the school, everybody got chickenpox. They get mild fever, leave your blisters after 4 days, 5 days, take a shower, and then you can go back to school. They are immune for lifetime. But some diseases, it is short period of time. We can explain reason for that, but at this moment, it's not necessary to understand or learn why it is that some diseases lifelong, some diseases a short period of time. But generally, no one can explain for specifically for certain diseases why is that. Better certain vaccines like we got in vaccination national vaccination program, then it's done. Certain vaccine you have to give the booster dose. I think, for example, even there is a vaccine around 14 and 15 years age. They have to take this vaccine. Other than that, after 15, 16, we don't take any vaccine as a regular program. But we can always take vaccine at any age if there is a risk of getting it. For example, let's say tourist. If tourist is coming from one country to another country, and he can check what are the diseases common in this country, and is the vaccine available, he can take the vaccine before his tour. Then it's done. But here, tetanus-like thing, we cannot give a guarantee that immunity is lifelong. We don't take the risk. Why is that? Look at this picture. Even if we neglect, and if we get the tetanus, okay,